So, all right, now you know already what is coming, but it was coming in a sort of a nice way. Huh? I wanted to show you the evolution of the universe. Here it is. And as I indicated, let me quickly go through again. It started 13.7 billion years ago with a big explosion. We leave to physicists the explanation of that. And then the age of physics started, where there were no atoms. Particles formed very quickly. Then atoms formed, atoms aggregated. And after the age of physics, the age of chemistry started around here. When atoms could form, when atoms could then connect to make molecules, molecules became more and more complex at making aggregates, defining compartments, inside, outsides, primitive cells, as we might call it now. And then something happened, which we will understand someday. We don't know it for the moment yet, but a new property appeared in this matter. After the age of chemistry, life started around this point, and the age of biology started. It continued to evolve, and the organisms, the biological organisms, continued to become more complex until a property appeared, which is thinking, represented by the thinker of Rodin. And this is the path towards complex matter, from initially no matter, only energy, to complex forms of matter, of which we are one complex form, and we are not the only ones in the universe. I'm convinced of that. And also we are not right now the way we will not be the end of the evolution, and it will continue. Now, we can then characterize this evolution by parameters, complexity, information as a function of time. It started with divided matter, particles. This became then condensed. Things went together, making larger entities, molecules, and aggregates of molecules. It began to be organized, then at some stage living, thinking, and maybe something beyond. But that's a bit difficult to know what that can be. There can well be something beyond. We cannot say it cannot exist. We just don't know. And in fact, thinking beyond your thinking, that's not an easy thing to do. But who knows? You have to leave things open. So that's the evolution towards complex matter. Now there's a big question then one can ask. What I consider really a very big, maybe the biggest question. How does matter become complex? What are the mechanisms by which matter becomes complex? The mechanisms by which it can lead to us or to other entities around the universe which are of the type of thinking organisms like human beings or of other types of thinking organisms. How can we go from the elementary particle to the thinking organism? And are there perhaps even higher forms of complex matter? For this, mankind has invented something called science. Physicists study the laws of the universe, the laws on which everything depends. Biology, chemistry, everything depends on these laws. These are the basic laws which regulate our universe. Biologists in this well-known fantastic center, among others, but special one, biology tries to study the rules of life. They're not laws, they're rules of life. The laws are the physics. These are the rules of life. And chemistry tries to build a bridge. Chemistry tries to understand and to provide answers to the question, how do you get from general laws to a specific expression of these laws, like living organisms on the planet Earth? What is the pathway to get to these entities from the general laws? General laws don't tell you what is the end of it. And this is, of course, then something chemistry can try to bridge from the general laws to their specific expression in a given part of the universe, like our planet. The answer to that question is, it happened by self-organization. Now, this, of course, you can see that's not an answer. It's a It's a word, sure, but we understand what it means. It happened on the basis of the laws of the universe, but it doesn't tell you how it happened. So defining it by a word makes us conscious of what is the process. The universe has laws which lead to self-organization, and our universe is built in such a way that it leads to it. And for Christian de Duve, I talked a lot with him, and we agreed that in fact it is even, one could say, a cosmic imperative. Our universe is built in such a way that life is not an accident. Life is an imperative. This can be discussed. That's also a philosophical question, but it's, I think, mostly a scientific question which we have to answer in the future. So before biological evolution existed, the one we know about, the one which has been called the Darwinian evolution, there must have been an evolution which had nothing to do with life. Because it's hard to imagine that you start from particles and jump right away into a biological evolution. So there was a process by which matter became more and more complex even before it was living. Because you have to go to the threshold of this property we call life. So there is a much broader evolution than the Darwinian, which is the chemical evolution. And even the Darwinian, we know, is dependent on processes which are based on chemistry, as we'll see in a moment. And you know that very well in this place here. Now, in this explanation of what we are doing around here, in this invention of mankind by curiosity or science, chemistry deals with the structure of matter and the transformation. Structure, how it is built. And transformation, that is something very important. And in fact, it attracted me to chemistry. The fact that you can act on the intimate structure of material objects. Then you can transform them into others. Then you can act on them. Then you can make new types of molecular objects, or objects making up our matter. Now, this matter is built on bricks, and these bricks are the elements. And these elements were progressively uncovered, put into tables, but it was rather chaotic until it was realized that there was an order in these bricks constituting our universe. And that was mainly due to Dmitri Mendeleev. He wrote in 1869 what I consider as being to be one of the most important papers in all of science of all times. In 1869, he published this paper which put order into the elements which had been discovered. That there were regularities, that you can put these elements which people knew looked like a zoo, like a chaos, that you can put order, that you can organize them. That this was number one. Here are lines. This is his original paper. He went a step further, however. There were also places where he put question marks because he think there should be something there, and that was found. That was discovered later on. This is what we now call the periodic table of the elements, which now looks like this. Now, these are from a hydrogen. These are, of course, also now, as we know, some artificial elements. But the important thing is, this table organizes the elements which make up our universe. Now, you should realize what that means. This little beast on this planet Earth has been able to generate a table which tells you what visible matter is made of in all regions of our universe. Huh? That means anywhere in the universe. If there are organisms made of our visible matter, they will be made of this. Why? It's a bit a problem for me, because scientists should not be dogmatic. A scientist should not say, that's it. But here it's very difficult to escape. Why? Because it's just like arithmetics. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. There's nothing between 1 and 2, 2 and 3, 3 and 4, 4 and 5. What are you doing here? You're just counting. hydrogen, 1 proton in the nucleus, 1 electron around it. Helium, 2 protons, 2 electrons, 3, 4, 5, 6, and so on. So you just build up your table by taking the particles and you make the elements. So this is, for me, something absolutely incredible, that we on our planet have been able to define what everything of visible matter, I leave the dark matter a question to the physicists. We don't know yet what that is. But the visible matter we are made of is made of that anywhere. And this is the playground of chemistry. Chemists like to use these pieces like Lego, like big children, Lego sets with these elements, putting them together, making new distributions, changing them around, and playing with these basic bricks and trying to generate out of them everything one can generate, which is, of course, an enormous number of things, of combinations. So, from the atom to the molecule, one has developed over the years what is called molecular chemistry. This is the art and science of linking together the bricks, the atoms, to make molecules, the houses, and by using strong bonds. The strong bonds are called by chemists covalent bonds. The strong bonds which hold these things together. Now, some milestones to show how this has happened progressively. In 1828, Friedrich Wöhler in Germany synthesized urea. He made urea from, which is a molecule contained in living systems, he made it from what is a mineral, so to say, ammonium cyanate. That was very important. First of all, he showed one of those chemical transformations. Let's make urea from ammonium cyanate. But even as importantly and more broadly important, at that time of Wöhler, it was thought that if you want to produce in the laboratory a substance which is contained in a living organism, you cannot do it without the help of a magic force called the vital force. By showing that it is not at all the case, you can make it in the lab, there was no vital force. That was very important also the way you look at what's around. What is life? What are molecules making life? What is life made of? There's no conceptual difference between the molecules of the non-living world and those of the living world. About 150 years later, chemists had acquired enough power over matter, so to say, to synthesize that thing, vitamin B12. That's, of course, an enormously complicated molecule compared to urea. It was done by the combined efforts of two groups, Robert Burns-Woodward at Harvard and Albert Eschenmoser at the ETH in Zurich. And this required, of course, a lot of work. There were about 90 steps to build it up, to put the atoms in the right position everywhere. And something like 150 man and woman years altogether, over 10 years, maybe even more than that. I was a postdoc with Woodward at that time. I made this. That's my part here. That's also interesting, you know, when you are a scientist, you contribute to an enormous project. And I think we should be proud of that, that we bring a stone. And if the stone is a small or a big one, but we bring a stone. That's our stone. And I sometimes, quite often, speak also to high school students. I think it's quite important to tell them that when you are a scientist, you contribute to an enormous adventure of trying to explain what we are, where we are coming from, and what the universe is. Now, after B12 was synthesized, of course, many more complex molecules even were made by various labs around the world. And molecular chemistry has very much evolved. And nowadays, one would not make it by the same, probably not by the same methods which were used in the 70s of last century. So, molecular chemistry has progressed and will progress, and there's still a lot of new reactions to be discovered, new molecules to be made, new properties to be found, new materials, new drugs, and so on. But then one can ask a question. Now this is something which is made for you in this place. This is a cancer cell, these are killer cells. The killer cells are supposed to go around, find out the cancer cells and kill them. How do they know? How do the killer cells know the other one is a cancer cell? These are white blood cells, blue dots, HIV virus. When the HIV virus hits a white blood cell, it can infect. How does the virus know it has reached the goal? Now these objects, biological objects, are nevertheless based on molecules. And they are defined by a membrane. And this membrane is like a soap bubble. It's molecules. And in this membrane, there's things sticking, which tell one another what the other guy is. So this process indicates that there's something happening between the molecules, meaning the objects, and not just inside. That leads to the idea that there must be a chemistry which is not the chemistry only of the molecular systems, what is in a molecule, but what happens between molecules. And this is what we then call supramolecular chemistry, because it deals with what happens beyond the molecule, beyond the separated molecules. When they get together, they attract each other, they repel each other, they hate each other, they like each other, they transform each other, and so on. And this is then going a step further and trying to understand and to manipulate not only the covalent bonds linking atoms in molecules, which is the art and science of molecular chemistry, but also building up an art and science, so to say, of this non-covalent chemistry where the bonding is what happens between molecules. Three main functions have been studied over the years: how molecules recognize each other, how they react on each other, how they can carry each other through a barrier, a membrane, like a cell membrane or an artificial membrane. Recognition is the most basic of these processes. That is what happens between molecules when they encounter each other. Now, what is this molecular recognition, which is this most basic phenomenon? First of all, it requires an interaction. If they don't interact, they ignore each other. There needs to be something, some way of talking to each other. There has to be some interaction, a binding. But this is quite normal. There's a physical force. But there's something else which is very important. When you go around the street and have to recognize somebody, what you need, information about the person. So, molecular recognition, recognition between molecules implies, in addition to the binding, also an information. So that molecules attach to one another in a specific fashion. In other words, they sort of recognize each other. They touch each other and they recognize each other. A very simple way of expressing that is that there is a double complementarity. I make it very simple here, but sort of general principles: geometrical complementarity and interactional. Interaction is very simple. Plus attracts minus, plus repels plus, and so on. Now, in fact, there was already very early on by one of the greatest chemists who ever existed, Emil Fischer. He proposed, and I guess all biologists must know that, in 1894, a very famous paper where he said for an enzyme to act on its substrate, they have to fit together like a lock and a key. This is, of course, nowadays, it's a bit more complicated. We know that locks are a bit soft, they can adjust, the keys can adjust. But in fact, it's a very basic, strong image. And this, again, in general public lectures, if people get away from my lecture and remember that what's happening in our body is a question of locks and keys fitting together, that's already quite something, and quite advanced even in some ways. So, fitting locks with keys, that's molecules fitting together and recognizing each other. In a very mechanical way, of course. That was the age of mechanics when Emil Fischer proposed that. We now know it's more complicated, but I think it's a basic, very nice and strong image. One of those images which define a science also and make it understandable to a very much broader public. Now, of course, I don't want to insist much of that, because that's the kind of thing, I guess, which at the Karolinska you see every day and every time, and you have heard it ad nauseam, I would even say. The fact, but however, it's interesting because that's the most basic information processing system. If we look at it in that way, it's a long string where you write information with just 4 chemical letters. And these are even simple chemical letters. Adenine, guanine, thymine, cytosine, 4 groups, heterocyclic groups. And these groups, they write the genome. It's just 4 letters. And that makes a difference between an elephant and a bacterium, and a tomato and a human being. Now, this long strand, as you know, wraps around each other, and so on. And this is the storage. But you have to read that information. You have to read it, and you read it by a binary system. As you know, I'm sure, the fact that it can bind together by 2 or 3 points of interaction. 2 or 3 points of interaction, which is binary. So you have 4 letters, 2 points of interaction. Why this? Because probably evolution started when it was good enough. Of course, you can invent other letters. You could have more than 3. You could have 4 points of interaction. Why not? But 4 letters and 2 ways of interacting is enough. You don't need more. People are now, some chemists are trying to introduce more letters, new letters, and so on, but you don't need it. In principle, it's enough. And in fact, in a tautology, the fact that it is enough is shown by the fact that we exist. So these are these hydrogen bonds, and this means also that the storage of the information is in the molecule, and the reading is on the interaction between molecular groups. So there is a molecular storage of information and a supramolecular processing of that information. So chemistry is an information science also, like biology, of course, but biology is based on molecules, the science of informed matter. My initial motivations for the area we have been developing were the following. I was interested in the functioning of a nervous system. You might say, what is this about? That is not chemistry. Yeah, it was not chemistry, but I was interested in that. And as a chemist, I was wondering how I can get into the functioning of the nervous system. Either you start your studies again, that's not very efficient, and you try to become a neuroscientist, which was a bit too early probably at that time, but it's still a fantastic field, of course. So I thought, okay, what could be an entry of a chemist into that fantastic goal which we have to try to understand? The propagation of nerve influx. Why? Because it's simple in what is doing the propagation. Just as change, the action potential is a change in sodium potassium ions across membranes. So there must be in the membrane, in the axon of the nerve, there must be molecules which are able to make the difference between sodium and potassium. Sodium, potassium, simple chemical objects. So the question came up, how can you make the difference between a sodium and the potassium? 2 little spheres with a slightly different radius. How to do that? And can you then do this, recognize sodium and potassium, and maybe understand the selective transport through membranes and their elective way of carrying them along? So, using the image of Emil Fischer, let's just be simple. If you have 3 keys, 1 lock obviously is the red key which fits. Then what are the simplest keys in the periodic table of these elements? The simplest object in 3-dimensional space is a sphere. What are the spheres? The spheres are the alkali ions. These are spherical objects with slightly different radius and with 1 positive charge. Lithium, sodium, potassium, rubidium, cesium, and in the middle here are sodium and potassium. These are the ones which are important for living organisms. These are the ones which make the transmission, the action potential. So it was an interesting problem, which was one of how can you perform spherical molecular recognition. Again, if you want to build then a lock for these keys, what should you do? You should make cages which have an inside, which has a shape compatible with a sphere, and which you can adjust in size. These are then the scriptants which were mentioned in the introduction. A crypt is a cavity. So building molecular cavities of adjustable size, and this was done many years ago now. This is the smaller, a larger, and chemists can do that in the laboratory. They synthesize these molecules. They can make small cavities, larger and larger, and these were designed in such a way that the first one is just compatible with the size of lithium, the second one with sodium, and the third one with potassium. So this was the start of trying to generate molecular systems which act as, so to say, a receptor for a given entity which will bind to it, which you may call the substrate, simplest case being spheres, but of course many more complicated and more complicated objects have then been studied. And around the world there have been many, this field of molecular recognition in artificial systems. Of course, in biological systems, it's obviously that is very important. In artificial systems, it is designing locks for keys or keys for locks. So it developed, and it would be absolutely impossible even to summarize that in talks for a week long or further on. But then you may also ask the question, what can be possible applications in life sciences? First of all, making locks for keys, or especially keys for locks, making artificial entities which will bind to a biological lock to either inhibit it or to amplify its activity. This is what you might call drug discovery. What is a drug? It's a key made, either found in nature sometimes from plants, usually from plants, or synthesized in the laboratory, which will fit a biological lock. We also, just to give you examples, of course I could give you many more, have developed another feature which is to use them for medical diagnostics, for gene transfer, and I want to give you also an application in biomaterials. We have been working on, this is just, I cite example from our work. Of course, drugs are all over the place, and making keys for the locks, this is what pharmaceutical companies are doing. That's what they're supposed to do. We have been working for many years on a substance which has a dual action. This substance derived from a natural product, it's myoinositol trispyrophosphate, acts as an allosteric effector of hemoglobin and makes hemoglobin release more oxygen than normal. It was also found that it is also inhibitor of a kinase, PI3 kinase, and an activator of PTEN at the same time, it is phosphatase. And it acts, therefore, on signaling pathways in tumorigenesis. I cite that because it's right now in phase 2 in clinic in Zurich. And to give you just an example of its action in animals, implanting hepatocellular carcinoma, that means it was Morris hepatoma in rats. So I give you just the comparison between the controls, which in 7 to 12 weeks were all dead. Treatment is doxorubicin, which is a drug which is used as in chemotherapy. It's about, they hold on a little bit longer. And if you treat them with the compound, they survive, all of them. And there is eradication of the implanted tumors. Now, there's still a lot to do. One has always been healing rats. One has been quite good at making rats healthy. We want now to see also if this is valid for humans. I'm going to just to give that example because it's one of those cases where this chemical key binds to a biological object, hemoglobin, and hemoglobin in red blood cells, and also to enzymes, and has these biological effects. The other example has to do with making objects which can be used as a sort of a nanobulb, a sort of a marker, a label for immunoglobulins, proteins and attaching those to immunoproteins has been done by Gérard Matisse and a small company in France, producing at the end of the game an apparatus which uses this cryptate of an ion called europium, which is part of the periodic table of Mendeleev, and which emits red light. So this absorbs UV light in its skin, so to say, in this molecule, transfers the energy to the europium, which is then released as red light, and that serves as the marker, the label for this technology for immunodiagnostics. The last example I would like to give here has to do with gene transfer. Why? Because if you can recognize an entity and help it carry it into a cell, that's a transport process. And of course, this was one of the basic functions which were studied, but it became biologically important when it was realized that you could, or when there was as goal to try to put a piece of DNA, that means a gene, into a cell and making express the corresponding protein. The first step is to enter it into the cell. For that, you have to try to build up a synthetic vector which recognizes that piece and then helps it to go through the membrane into the cytoplasm. And then, of course, a lot of complicated things happen afterwards. I don't want to get into that, of course. And then, at the end, expressing the final protein. This has been done also using basic principles of supramolecular interactions, whereby one is able to facilitate the entry of DNA into cells. Of course, there are other techniques which use viruses, viral-derived technologies, but this is a purely synthetically derived technology, which is of interest for biotechnology, to make genetically modified organisms, and to have some specific genes expressed in bacteria, but in animals, and also gene therapy, for instance, for cystic fibrosis, which is a disease of a genetic disease due to the malfunctioning of a chloride channel in the lungs, and where one can hope to transfect yourself just by inhalation of the transfecting agent. There are some examples I could show that, where you can transfect the epithelium of the airways of a mouse, but I wanted just to give you the idea that gene transfer is a transport process which can be facilitated by this type of technology. Now, the next step, once you know a bit more about molecular recognition, you would like to use the basic features of recognition to generate systems which would be able to self-organize. That means designing the bricks and the pieces so that they can get together and lead to a complex object at the end. You want that to happen spontaneously on the basis on which these are built, but you want to control it. You want to control it by the way you design the object, the objects, and the way they have to get together. Now, of course, one example, and I could have cited many others, for instance, the tobacco mosaic virus is a case like that. That's a bunch of molecules which go together on the basis of the way the pieces interact. 2,130 bricks, and there's a viral DNA, and these bricks go together by surface recognition of the pieces, and there's nothing magic. It happens automatically, but it can all be explained by structural chemistry, organic chemistry, physical chemistry. It's nothing magic. It just happens like that. Because these basic bricks, these proteins, are constructed the right way so that they recognize one another surface to surface. In fact, there's something you might be interested in, because it looks sort of magic, this kind of self-organization. But there is a video on, you can find it on the internet, where people, I think it was in California, they did the following. There are some spherical viruses which are made from a concave type of proteins. So they, on the macroscopic scale, they took pieces of plastic which, when you put them together, the correct number, would form a sphere. They put on these pieces of plastic little magnets, which would interact with one another at the right positions where the proteins do that. Then you put the whole thing in a big beaker and you shake. And after 2-3 minutes, you have a sphere. Which means it's all built into the construction, in the way they interact, and you get it at the end. They push it even further. They made a mixture of objects, red color, of a dimension somewhat larger than other similar object, but a bit smaller, green color. Then you have to shake longer. They build a machine, a shaker. But after about 10-15 minutes, you have a red sphere and a green sphere. Nothing magic. It's just the basis of the way matter organizes, the way in which things want to get together because they bind to each other, where the binding is driving the system to generate the objects which have the strongest binding, the best binding. So this is like a programmed chemical system, this virus, where the program is molecular. The pieces which make up the virus, the proteins, contain the molecular program, and the operation is supramolecular, the way they interact. So the storage, comes back to what I said about DNA also, the storage of the information is molecular. The operation, the reading, the processing is on the supramolecular level. You enter the information, then it works by interaction with one another, and at the end of it, you terminate the system, and you have the final object. That's sort of a self-organization programmed on the basis of how your objects are constituted. Let me just then give you an example which has nothing to do with anything biological, except that structurally it has a little bit of, make you think about some biological objects. And here we will do the following. We'll take bricks, which are molecules, and these molecules are designed in such a way that they can bind a metal ion. So the bricks are molecules. The cement, like when you build a house, the way to cement the bricks together are metal ions, copper or iron or nickel and so on. And so what can you do? You can make double helices, which have nothing to do with DNA. This is an organic molecule which has been built on 3 subunits called bipyridines. And that you can say this is a trinucleotide, like a trinucleotide, except that it has nothing to do with a trinucleotide. And you can bind now copper 1 to these sites. And this is designed in such a way, I don't want to get in details, I have no time, so that they will wrap around each other to form a double helix. This is not just a nice picture, that's a crystal structure, that's the way it looks. That's the correct double helix you get from making this, adding copper 1, and spontaneously you get a double helix. Similarly, you can make a triple helix by changing a little bit the strand, adding another metal ion. In this case it's nickel, or it could have been iron or cobalt, and you get a triple helix, again the crystal structure. For this I just want to show you that you can design chemical objects which have nothing to do with anything biological, into which you put the structural characteristics, which you might call the information, necessary, so that on interaction with the cementing unit, in this case the midline, will automatically lead to the final object. In fact, I could have shown you many more than that. This is also now an area which has enormously developed, where many groups around the world have made more and more complicated pieces, putting them together with various metal ions and achieving synthetic architectures which otherwise you wouldn't have thought would be possible. 15 years ago, people would have said, yeah, you are mad. You will never get this. And it forms spontaneously. You just mix and you get. But you understand why you get, because you design your brick, you use the right cementing unit, which brings it together in a given geometry, and at the end of the game, you get the object. Now you can ask why? Because thermodynamics rules the world, and you get the most stable object. Now, this is a very, very brief illustration of the kind of self-organization which you can produce in the laboratory with synthetic entities by design. You make the bricks, you put in, so to say, the information, the structure information, the interaction information. You sort of program the system, and it will run along and generate a final architecture. But a new thing, a new twist, it has appeared more recently then, because after all, self-organization by design, you try to get to the goal which you have put for yourself. But it would also be interesting to go a step further and to do this artificial self-organization with selection, a selection process. Selection meaning that you will have a diversity of objects, but the process will select among this diversity of objects the one it needs to generate the final entity. For this you need, as I said, diversity, different types of pieces so that the system can choose what it needs, but you need also dynamics. That means you need the objects to be labile. You don't want them to be stuck at some point. And we know very well that in biology everything is labile. If we were concrete, it's not living very much. Huh? So we are labile objects because things get together, they find out how it works, they find the right position. You don't want to be stuck. And so this is also a feature which has to be introduced. If that is possible, then of course you can also hope that on changing the conditions, the object in question and the process in question will adapt, and that opens the way to what we now are very much interested in, to adaptive chemistry, a chemistry whose object will adapt to what happens around it in the environment. Let me indicate to you how this can be sort of approached. I showed you earlier these entities, the strand and the metal ion, which were giving double helices. Now we will mix them with the ones which give triple helices. Now we have a diversity. And we also know that these objects can exchange. They can get together, dissociate again, reform. So the question then is, which is quite interesting, whereas when these entities are isolated, we know which gives a double helix, we know which give a triple helix. Now if you mix them, will it still do the same? It does. That means that is a system which, in the diversity of objects, selects what it needs, and that the mixture generates what separate entities would generate also when separate. And this is then a process where it's a selection process where this organization of the chemical objects generates the right objects despite the fact that they are mixed now. And that is, of course, also in chemistry quite a step, because usually in chemistry you want to produce entities, single entities, and you usually don't like mixtures. But one can, of course, claim, and for biologists, there's no need for a long explanation, that there's much more interest in mixtures, provided they do what they are supposed to do. And for instance, the best illustration, take a cell. What is a cell? It's a horrible mixture of many, many things, but it functions, it works. Because the molecules know what to do, they get together, they recognize each other, they do the right thing at the right time, in the right place. And this is also a path which chemistry must follow, to create complex systems where this kind of complicated features can be present, not necessarily with biological entities, because the world is much larger than the molecules of biology. So this is what we now call a dynamic chemistry. Dynamic in the constitution of the object. The supramolecular ones, because the binding is weakish, labile, are intrinsically dynamic. That means they can indeed dissociate and reassociate, maybe differently, maybe incorporating other pieces. And this is a non-covalent dynamic, but you can also introduce that into molecules. In as much as you make molecules where the linkage between the atoms are made fragile, are made be made dissociable. In other words, you can break them and reassociate and break and reassociate. In other words, you make also molecules which contain linkages which can be broken and reformed so as to be able also to exchange their components. And we now call this a dynamic chemistry which deals with the constitution of the chemical object. In other words, the chemical object is not anymore static, but it can exchange its pieces, either on the supramolecular level, when the interactions are the non-covalent ones, or at the molecular level, when the reactions which form the bonds are reversible. They can dissociate and reassociate. And that opens the possibility then of adaptation. So this means sort of that starting with molecular chemistry, building a supramolecular chemistry on molecular chemistry. This supramolecular chemistry is a dynamic one, and then you introduce the features of supramolecular chemistry back into molecular chemistry, which opens the way to this dynamic and adaptive chemistry. A sort of Back to the Future, like that movie. You go for molecular, supramolecular, you go back, put it in, and the future opens. So what is it good for, this type of chemistry? There are many things. I can only illustrate a few. First of all, you get a soup, a mixture. But mixtures are interesting. Mixtures have diversity. Mixtures are rich. So you get dynamic libraries, dynamic sets of entities which have a lot of features by the simple fact that they can exchange and can recompose their components. You create, therefore, diversity, and then you can operate selection. If you have diversity, you can have selection. So I would like just to give you examples of two things. On one hand, can you use this type of approach for discovering biologically active substances? Can one make dynamic nanostructures? But this I will not discuss. Can one make dynamic materials? Materials which would not be static materials, like the ones like this shirt, but dynamic ones. What about applying this kind of approach to the discovery of biologically active substances? And here, to be simple, I use again the lock and key image of Emil Fischer. Ideally, you want to know the lock, you want to make the key, and you want them to fit. That sounds good. Then you understand molecular recognition. But you have to have the lock. In more biological or biophysical terms, you have to have the molecule, which is a lock, the protein. You have to get its structure. You have to know how it looks. You have to design the key. It has to fit, and then you're happy. But that is not so simple. Then came around people, here I simplify enormously, of course, who said, forget it, too long. We will make 1 million keys and try them all. We have now fast robotic, high throughput screening. We can screen, let's say, 100,000 a day. If the first 100,000 don't work, we make another 100,000. Okay, that may be perhaps more efficient, but it's not very attractive because you don't much understand how you get there. Of course, if you find something active, you can always try to understand afterwards. So that is a bit bothering, a bit the problem. But so what can one do? Design chemistry is the power of recognition, of getting the right information. Recombining objects is the power of combinatorics, creating diversity. So can one combine the recognition, the information, with the combinatorics? This can be done in principle if you develop a dynamic combinatorial chemistry. And just to illustrate that, let me give a picture which is easier to explain. You're now not looking for keys anymore. You just want a collection of components which have different shapes, different properties, and they have at the end a little functional group which makes them able to get together and to generate a whole set of dynamically interconverting combinations. In other words, in doing so, you have access to all the combinations of all the objects at any time. This is then a way to generate a dynamic set which has all these constituents always in equilibrium. Then you can, yes, sure, but what are you going to do afterwards? Not very complicated. We are doing thermodynamics. The law of mass action, Le Chatelier's principle, tells you what? That if you add now to this the lock will pick up the key which fits best, automatically. So, if you have this mixture of equilibrating entities all the time, when you add the lock, it will pick up the key, its best key, among the ones available. And this then is a screening which is done by the system itself, combining the features of combinatorics, generating high diversity, and the features of recognition of information, which is simply present in the interaction between the two, molecular recognition. Now, this looks like a panacea. Of course, in science, there is no panacea. It has features which make a problem. For instance, suppose you make that the library, which is only in the converting object, has 1,000 constituents. Then the binding to the final receptor has to be strong enough so that you can detect the very small amount of the one which is the good one in this mixture. But this has also an advantage, that if you see something, you know it's good. In other words, the fact that if you have a large library of objects where you need a strong binding so as to detect the very small amount, or 1 in 1,000, that is a problem perhaps, because you might not see anything. But if you see something, you know that this is now stable enough, and you have a good lead to develop then a drug. But of course, you need at the end of the game, you need some medicinal chemist who take this lead and say, okay, let's make it better, but we know where to go. And this has been applied to the discovery of enzyme inhibitors, to receptors of biological substrates, and so on. For instance, it can be interesting also, since it's reversible, for making dynamic drugs. That's a sort of a dream, you know. Dynamic drug delivery systems where you have a biologically active substance which binds to a support in a reversible fashion, so it can release it in a controlled way. And this, maybe in the future, can be interesting, because you can make cocktails of drugs which would be released at different speeds and then generate different activities along the way. Of course, there's also a regulatory problem, because you have to show that none of the combination is toxic. And the FDA might make some problems. But the principle is interesting, to make cocktails which will be progressively released. On a less biological scale, but still interesting one, in addition to the controlled release of drugs, we were looking at that with a perfume company to make controlled release of perfumes. And as you can make them release sequentially at different rates, you might smell the violet in the morning and the rose in the evening. For the moment, it hasn't crossed yet into commercial activities, but we found that as sort of an interesting possibility. Now, what about materials? I told you also that one can think of using these dynamic properties for materials. And these materials would then have an interesting property. They, especially when they are polymers, dynamic polymers, they could become self-healing, they could become adaptive materials, they could have properties which static materials cannot have. I will just to illustrate that. Of course, they can be either molecular dynamic or supramolecular dynamic. And I would like to illustrate both of them just by one example each. First of all, if these polymeric entities are made of components linked together by non-covalent bonds and making then this polymeric molecule. Then you define, in doing so, a chemistry which leads to a supramolecular polymer chemistry as compared to the normal molecular one, the one which makes our shirts, nylon, and so on. Now, this was interesting. We introduced that in 1990, as you can see here. And this is an illustration of also how long it takes for a basic proposal in basic research to come to an application. 1990, okay? 23 years later, in 2013, a company had developed these principles of supramolecular polymers for cardiovascular implants. Let me just illustrate that. Children having a congenital cardiac malformation need cardiac reconstruction. And there's a small company, Xeltis, which has developed cardiovascular implants based on supramolecular polymers. They were introduced into man, in that case children, by Professor Leo Bokeria at the Bakulev Scientific Center for Cardiovascular Surgery in Moscow. I did not know him, but I now know that he's a very famous pediatric cardiac surgeon and was introduced into this young girl, Dominika, on 23rd of October 2013. This is Bokeria. He looks like a surgeon, doesn't he? Here's little Dominika. She's fine. This was 3 months checkup, but now it's more than a year. And it was introduced, 10 children altogether, and it's continuing, and it seems to work. Now this also illustrates, first of all, that it's difficult to predict where what you are doing will end up. For getting from the proposal of a supramolecular chemistry to cardiovascular implants in a child, how do you find it out? Because on the way, there's to be people who detect that it may be interesting. Then people who make the material. Then you have to make the implant. You have to test it on animals first. Then you have to find the surgeon who is ready to do the job. And of course, you have to, along the way, to have the finances to do it. Not evident. And so, okay, I guess I don't need to convince you. I usually say that you need basic research, blue sky research, opening new possibilities, and some of those new possibilities, maybe more than what we think, but we don't know when, will lead to interesting, very useful applications. So another illustration deals now with sort of what you would call normal material, not biomaterials. And this is relying on the following property. Suppose you make a film, plastic film, but a plastic film which is a dynamic one, where the objects which make it AB, AB, AB are objects which can break apart and reform and break apart. And you make another film, A prime, B prime, where A and A prime are the same category of molecules, B and B prime are the same category, but different molecules, but of the same category. For instance, amines, aldehydes, carbonyls, and so on. You superimpose. Obviously, if you have AB and A prime, B prime, two new combinations can appear. A can combine with B prime, because they're just in contact, and A prime can combine with B, meaning that if this bond is dynamic, can break, form, and dissociate, you will get this. Two new combinations appearing. If these two new combinations have an interesting property, you will generate it in this way. Let's look at a case of demonstrating that by something visible. It's always better demonstration if you can see it. Suppose the film AB is colorless, not fluorescent. The film A prime, B prime has maybe a little bit of color, a little bit of fluorescence. You superimpose. At the place where you superimpose them, obviously, new combinations will appear. A, B prime and A prime B. A, B prime, A prime B. If one of these two new combinations has a color or fluorescence, you will see it. Okay? So, this was done in collaboration with a Japanese company, Mitsui Chemicals, and they were sending collaborators to my group in Strasbourg. We were developing the basic science, and they were looking whether or not they had applications. So, here is the illustration. A cat where the head and the ears is polymer AB. The mustache, the eyes, and the inside of the ears is polymer A prime B prime. You superimpose. You heat to accelerate a bit the reaction. They heated a bit high, I must say, but they wanted to have the result quickly. Color, fluorescence. In other words, the recombination has occurred, has generated the other two possibilities, and one of them was colored and fluorescent. So that proves that it works. Two, that you might be able to write with a heating laser at the interface between two films. And if you make a stack, a millefeuille of that, you can write in 3 dimensions, in the plane and up and down. So this is just an illustration of the possibilities offered by the materials which are not static, which of course do not have the same properties as static ones because they may dissociate, but they have properties which the statics do not have. The same can be done, and here I don't want to insist too long. The biological macromolecules can also be made dynamic. Of course, you have to modify them. You have to introduce a bonding which is dynamic. We can make dynamic nucleic acids, di-NAs, not DNAs, but di-NAs, dynamic nucleic acids, dynamic peptoids, analogs of peptides where the bonding, the amino acids, residues are bound together in a reversible fashion, and also dynamic sugars, dynamic polysaccharides, where the units are also bound together by reversible bonds, which makes them able to break apart and reform and adjust. So I would like to finish just, and I was told by Professor Engbert that there may be people in the audience interested in systems biology, just to give an idea where this type of dynamic chemistry leads into systems. I insist mostly on the principles, because the technology behind is a rather maybe complicated chemical way of doing things. So, adaptation, what is an adaptive system? It's a system where the entities which compose it can react to change in environment, change in phase, going from water to organic solvent, for instance, or going through a membrane, for instance. A physical effector, that can be heat, can be pressure, can be light. A chemical effector, protons, metal ions, other molecules, and even changes in shape. And with this, we can form a network. Let me just show you how these chemical adaptive systems can be generated. And for that, I will illustrate first, since we were talking about building up systems on the basis of chemical entities, how can we describe this? We will do the following. I go back to this simple case of just 4 entities, AA prime, BB prime, which can interact with each other or react with each other, AA prime with BB prime, thus generated AB, AB prime, A prime B, and A prime B prime. No, the 4 combinations, okay? Now, what network can we construct with this? We can make a square. We have 4 entities, so we have 4 corners. AB, AB prime, A prime B prime, A prime B. Now, the interesting thing is you can define the relationships between these constituents. On the edges, the constituents share a component. AB shares A with AB prime and B with A prime B. These are antagonists. If AB increases, AB prime must decrease, because AB will pick up the A from here and the B from here when it increases. On the other hand, on the diagonals, you have what you may call agonists. Agonists meaning that they do not share, they have no common component. And as they have no common component, they will do the same. When one increases, the other must increase. How? Easy to see. If AB increases, it will liberate B prime and liberate A prime, therefore making A prime B prime. So this is just a very simple example of a network where all the points of the network talk to each other. And we do something to one point, you will influence all the others. So you can therefore have networks which will react to changes occurring, and they will be adaptive. I give you first a simple example. It's unfortunately, I'm sorry, a very chemical one, but it might illustrate what's going on. Suppose you start with these four constituents. AB, AB prime, you see A is this unit here, it's again here. B is this unit 1, it's again here. So we have four objects which combine 2 by 2 and generate this mixture. Now, one of those happens to be a good binder of a metal ion. It's this one. So what you do, very simple, to demonstrate the principle, you add the metal ion. What will it do? It will bind to this one. But in doing so, it will perturb the whole setup and amplify this one by picking up that B and that A in addition. And as a consequence, the agonist will also be the only one, because the other ones, the mixed ones, will all disappear. So what does that mean? That means that you have an amplification induced by the metal ion, and if you replace it by a network, that's the network at the beginning. You add this effector, the metal ion, it will amplify this, but that leads to the amplification of the other one. And that then has interesting consequences. It's an adaptation process to the addition of M, the metal ion. The system reacts by adapting to the fact that you have added it. But that has also an interesting consequence: agonist amplification. In other words, the fittest for what you want to do, that means the fittest is this one, also amplifies the unfittest. That's an interesting, maybe in evolutionary biology, this could be something interesting. Amplifying the fittest necessarily leads to the amplification of the unfittest, because it provides a biological niche for the other guy. The fittest just picks up what it needs, but all the rest is left. So that's a very simple system, which sort of shows that you can indeed do this. And furthermore, if for some reason this unfittest has an interesting property, for instance, it's fluorescent, but you cannot make it directly, then you do it by acting here, and you will get the fluorescence, but from the other guy. Now, I have another case, which is a sort, I think since now we are in the principles, forget about the basics which are behind. But there's something interesting I would like to show you. It's a bit complicated, but I would like to show you that. These are, again, these exchanging entities, where you have lipophilic groups which like to be in organic solvent, and hydrophilic groups which like to be in water. Now you put them in a mixed solvent. And then you add something which separates into water and organics, okay? You make a phase separation like that. What will happen? They will redistribute so as to generate more of the lipophilic, more of the hydrophilic, which will spread between the two phases, okay? They will go up. The more the lipo will go up, amplify itself at the expense of these two here, of the mixed ones, and the hydro will go down in the water. And of course, you can reunite it. It's just technology. But the most interesting is that you separate and then you redistribute. So how can we now represent that by the network? Because there is a network thing I would like to show you. We start with a square, a two-dimensional network, when it is mixed, a single phase, like what I showed earlier. That's it. AB, AB prime, A prime, A by B. Then you separate. When you separate, you get a square in the organic phase and a square in the water phase. And you have now things talking to each other through the interface. This interface, exchanging things through the interface. What does that mean? That's quite interesting also when you think about what's going on. First of all, you have again, like in the square, an agonist amplification. Amplification of the fittest leads to amplification of the unfittest. But they are not anymore in the same phase. If you amplify, for instance, if AB is the fittest for whatever you do, it will amplify the other one, but downstairs. So they talk to each other through the interface. This means that you have an information which is presented in the distribution. It's not an information which is written like the, let's say, the DNA information. Here, the information is the distribution. And if you change the distribution, you have a different information. So it also means that you have a generation of signals and of a message without having a transmitter. In biology, we know that we have transmitters which carry a given information, for instance, from the nerve terminal to the head of the next nerve. Here, that's not the case. What happens is you change the distribution of your objects, and it's this change in distribution which contains the message, and if you modify something downstairs, you talk upstairs without going there. You transfer a message without sending a messenger, because they just redistribute. So these are these informed adaptive networks. I'm sorry, I hope the principles were clear, but of course there's things behind which show that they work. So, to conclude, adaptation through selection, yes, this constitutional dynamic chemistry allows for that, for the possibility to introduce into chemistry this notion of selection, making chemical objects which can undergo selection processes towards a chemistry which will become more and more an adaptive one, where the chemical objects you look for, is able to react to what is going on around. And of course can be extended to materials and technologies. So we start with molecular chemistry. Chemistry does not exist if there are not molecules at the start. You have to start on the basis of molecular chemistry. On this basis you can build this supramolecular chemistry I've indicated, designing the objects. Then dynamics, you introduce the dynamic features into both levels, ending up generating this, constructing this constitutional dynamic chemistry, and then you can operate selection and go towards an adaptive chemistry and hopefully an evolutive one. The difference I see is the following. The adaptive chemistry reacts to the external factors, but then you have to fix the characters which have been acquired to go a step further. We can do that chemically, but I wanted just to illustrate that. So, two final slides about how I consider chemistry to convince you that it's an interesting field. Chemistry deals with everything that can be made with the bricks of matter. Everything, of which biology is one expression. So, discovering what is around is, of course, very important. But chemistry can also create what is not yet around. So that the book of chemistry has to be written, not only to be read. The score of chemistry has to be composed, not only to be played. So there's a lot of creative power. That is what attracted me to chemistry, the fact that it can act on matter. And chemistry is, you might call, a Promethean science, an art of matter, like the artist brings out of a stone a sculpture which was not contained in it. The stone is just a stone. The same way, out of this periodic table of the elements, you can bring out all the combinations which don't exist yet. And I'm pretty sure the world is wide open to all these things which don't exist yet. And Maybe some of you are chemists and will contribute to it. Thank you very much.